Okay, so hold on to that specific question, and hopefully tomorrow I'll be able to have some examples for addressing it. Are you guys all able to see my screen?
Yep.
Okay, great. Let's go ahead and pick up right where we left off before lunch. So we were talking about how to build an RSEM index, and then after that we went ahead and submitted our jobs to count the number of genes and isoforms that align for our flight rev 1 sample. And so we took some time looking at that standard output from RSEM, noticed that we had several reads that got that warning about the seed length. And so that's that specific option in the RSEM documentation where we just scroll down. It's under the advanced options in the RSEM documentation for calculate expression, which is the RSEM function that we use to do our quantitation. And so the default setting, which is the setting that we use, is set to a length of 25. So this basically is giving us that warning, telling us that it's not going to count any reads that are shorter than length 25. And as you guys all mentioned in the chat, and we can recall from earlier on in the boot camp, when we did our trimming, we had our setting of 20 base pairs as our minimum. And then again, during the alignment base pair minimum was 20. And so that actually is less than the RSEM minimum. And so that's what happened is some of those reads that were in between, from 20 to 25 base pairs, they're not counted in the RSEM quantitation because they don't meet the low end cutoff for the default of RSEM. So of course, if we wanted to be consistent with those 20 base pairs throughout, we would have changed that to 20 in the options. And that might be something we might want to bring up to our AWGs when we revisit this pipeline, which we do every year to make sure we keep it up to date and make sure that we're generating data that's most consistent with the best practices currently, especially with bioinformatics, because this field moves at lightning speed. It just moves so fast. Any questions on everything that we went over this morning before we start taking a look at the GeneStar results file for our specific samples? Why was the seed length of 20? Yeah, great question, James. Honestly, I'm not entirely sure why we didn't change that default option. I didn't know enough about it when I started on GeneLab. I know a lot more about it now. And I'm surprised that none of our analysis working group members, many of whom have strong bioinformatics backgrounds, brought it up. So that's a great question, and it is something that I'm going to bring up the next time we revisit this pipeline, which is likely going to be later this year. So good question. We probably, in hindsight, should have made this setting to be consistent. Is there a way to check if the short reads are evenly distributed versus based to some genes? You mean evenly distributed amongst the genes that, I guess, I'm not sure I understand your question. Felix, you want to unmute your mic?
So the short, if all the 25 lengths are on one gene, extreme example, then this might affect your analysis. But if they're distributed over the genome.
Got you. Great question. Is there a way to check that? So yeah, you can check that. So what you can do is, if you guys remember, we were kind of playing around with our alignment files here. And so in that alignment files, we have an insert size, and we also have the exact read sequence that was mapped. And so that's our trimmed read sequence. So a couple of things we could play with, some Unix command, or for those of you who like Python, or pick your favorite language, we could play with some commands, since that sequence is always going to be in the same column of our BAM file. So that sequence is always going to be in column 10. So what we could do is take our BAM files and take column 10 and do a cutoff range so that we only take the rows that have in column 10 less than 20 base pairs. And then we can kind of subset our BAM file that way. And then we could take a look at that subsetted BAM file and then look at the gene ID or transcript ID in this case, and be able to know if all of those shorter reads were mapped to the same gene or not. That's a good point. Yeah, there's just, I mean, there's so much information in BAM files. There's a lot of things you could do, a lot of ways to play and troubleshoot and kind of really get a grasp on just how your data is being analyzed. And I love how you're thinking, just really thinking about how these nuances and how these settings that we put in the different tools can affect ultimately our outcome. And really thinking about making sure that we're not introducing biases in our computation, or at least minimizing the number of biases that we're introducing in our computation as much as possible. So I love the way you're thinking. Okay. Any other questions for anything that we've covered so far before we take a look at our genes.results file? Okay, are you guys all ready? Is there anyone who doesn't have their notebook open? Anyone who's not seeing Python 3 idle on the bottom here? Okay, great. So that means you guys are all here with me. So let's go ahead and take a look at our genes.results file from that flight 1 sample that we've been analyzing throughout the bootcamp. So you guys can go ahead and run this code block. And again, we're going to look just at the first few lines, specifically the first headlines, because we're using the head function of the Unix. We're using the head function in Unix, which we know by default prints out 10 lines. And so I want you guys to take a look at this genes.results file. I know I went over what each column is in the lecture. I also repeat the definition of each column in the Jupyter notebook. So you guys have that information in multiple locations. And then another link here to the description of the output files in RSEM. So taking a look at the first few lines, make my screen a little bit bigger here. So taking a look at those first 10 lines of this output file, I got a couple of questions for you guys. So what is the name of the first gene listed? And by name, I mean ensemble ID, of course. You guys go ahead and put your answers in the chat. Yeah, big shot, always super fast. Yeah, so it's the one that ends in 001 here. And for that first gene, what is the gene length is calculated by RSEM, for which I'll find examples of how that calculation happens before tomorrow. So looking at that first gene, what's going to be the gene length? Yeah, 3,262, and that's in base pairs. So that's 3,262. Well, technically bases, right? Bases long, since we're looking at transcripts, which is one strand. And what is the expected count of that first gene? So the expected count, remember, is the raw counts based on that, how RSEM does the calculation with doing the uniquely mapped, looking at uniquely mapped, and then based on those uniquely mapped, they use a maximum likelihood estimation to determine how to quantitate or how to distribute reads that are multi-mapped, and then they redo their summation for the quantitation for each gene based on that. Yeah, 869. Is correct. Kevin noticed he read the wrong column. Yeah. So you want to look at the expected count. I know it's kind of shifts, so you got to follow the tabs here. And again, you can always print out a specific column using different awk commands if you like awk, or of course you can do this in Python, because I know a lot of you guys are comfortable in Python, to pull out a certain column to make sure that you're reading things right. But yeah, it's 869 is the expected count based on that RSEM calculation for this first gene that we're seeing here. What is the count in transcripts per million, or TPM value, for that first gene? Yeah, 595, right. See a bunch of you guys put in that chat. Awesome. Now I want you to go ahead and tell me those same values for the third gene in the list. So first, tell me what that third gene in the list is. And then tell me what the gene length of that third gene is. Yeah. Yep, so that third gene ends in the 28, so we're looking at right here. And then we're looking at all those values there. So what is the gene length? Veronica, I think you're looking at the wrong line. So you're looking at the effective length. Yeah. Yeah, there you go. So the first column here, the first numeric value represents the length. And again, I'll get that calculation for you and how that's calculated tomorrow. And then that effective length is right next to it. So that was the one, Veronica, that you were looking at. And effective length is the one that takes into account the percent of each isoform that's expressed in your actual data. And then what about the expected count for that ensemble gene ending in 28? Yep, 91. So 91 is the effective count there. And then what about that count value in transcripts per million? Yeah, 1.15, right. Yeah, so as I mentioned before, RSEM spits out both quantitation based on gene IDs, and it also spits out quantitations for each transcript, so for all the isoforms. And so let's go ahead. You guys already answered all those questions. This is a little extra, but I think you guys should do it. So the GeneStat file, those expected counts are what we're going to be putting together in the counts table in the next step, our final step of this Jupyter notebook. But just so that way we can see what this isoform.results file looks like and how it relates to the GeneStat file, let's go ahead and take a look at the first 10 lines of that file. So you guys can go ahead and run that command. And again, this is for the same sample that we just looked at the GeneStat file. So I want you guys to see how this is set up. So as you notice, you'll see some columns that look pretty similar, just kind of in different orders. And so how this is broken down here is this first column now is your transcript ID. So you're going to see a lot more columns, of course, because there are genes that have multiple transcripts. And then in the second column now is where you'll find your respective gene ID. And so for transcripts that came from the same gene, that gene would just be repeated for each transcript that derives from it. And then after that, the rest of these columns should look exactly the same. So you're going to have length, except this time the length is going to be your transcript length. And then you're going to have your effective length, except this time it's going to be the effective length of the transcript. And then you're going to have your raw count data, or that maximum likelihood weighted count data that RSEM calculates, titled as expected count, followed by the two types of normalized count data that RSEM produces. And then it also gives you a third column here, or a third column, an additional column at the end here. And so that additional column, you see it says ISO PCT, that stands for isoform percentage. And so based on the data that you provided, so based on those reads and how they align to the different isoforms, it gives you a calculation of what percentage of that isoform is expressed. So what percentage of that isoform is going to be expressed? For genes that express multiple isoforms, of course, it's going to be some fraction of 100, which should total 100. And if there is no isoforms for that gene, that iso percent should be 100, right? So for this first transcript here, it's the only transcripts that's derived from this gene. And so the percentage of this isoform, it's the only one that exists, so it's 100%. Whereas some of these other ones here, right, like this one, we see that there is a breakdown. So there's 4 different isoforms in gene that ends in 28. And so you'll see the breakdown of the percentage of expression of each one of those isoforms and how many of those 4 isoforms are expressed in this sample for gene 28. You guys can write your answer in the chat. So there are 4 isoforms for this gene, but how many of those 4 isoforms are expressed? Yeah, Felix, Leah, you guys got it right. So 3 out of the 4. So we're looking at the count column for that specific isoform, the expected count, and we see that it's 0. And then you can also tell too, because the percentage of this isoform that's expressed is 0. So that means that there were no reads that aligned to that isoform of that gene. Yeah. So I want you guys to take a look at this and answer some questions. And just so you know, when you're looking at the gene effective length, that's where that isoform percentage is used when it weights how to determine the length. So again, the isoforms are going to be different lengths, right? Because they have a different number of exons. And so what exon, the weight of the length of each isoform is going to be dependent on its expression to give you that effective length in your genes.results file. So that's how it's calculated with that iso percent. Okay, so taking a look at this isoforms.results file, got a couple of questions down here for you. So the first thing I want you to do is I want you to calculate the sum of the expected count values for all isoforms associated with the gene that ends in 28. So with this gene right here that we were just talking about. So what is the sum of the expected counts for all of those transcripts that are derived from gene that ends in 28?
Could you reword that question?
Yeah, of course. So what I want to ask here, and Dick should give the right answer, is so this gene 28, it has 4 different isoforms. And isoforms are represented as different transcripts. So 4 different transcripts, each one of these transcripts are an isoform of gene 28. And so what I was asking is, what is the counts? So how many reads in total are there for gene 28? And so how you would do that is you would add up the expected count column for each one of those isoforms of that gene. So I'm looking for the sum of the number of reads that are expressed in each isoform of gene 28. Well, gene that ends in 28. Does that make sense?
Which one are you looking at under the expected count? Is it the 1943.54 or 74.7?
Yeah, so I know it's a little hard to see the things here. So your first column here is going to be your transcript ID. Your second column is going to be your gene ID. Your third column is the transcript length. Your fourth column here is going to be the effective transcript length. And your fifth column here is going to be your expected count. So looking for that fifth column, which is your expected count. So those are the count values for each isoform. So count being the weighted number of reads based on RSEM's maximum likelihood estimations. Does that make sense? Is it still not clear, my question? Yeah, I figured. Yeah, so I know it's kind of hard again. because of how these are tab separated columns. And so based on however long each value is here or string, it will push the columns so they don't like exactly line up looking at it like this. If you were to open this in like an Excel file or something like that, then of course everything would be nice and aligned. Yeah, so I see a lot of you guys have the right answer here. So it's going to be 74.7 is the expected count value for this first isoform listed, followed by 10.19 for the next one listed, then 6.11 for the third transcript for this gene listed. And then the last transcript is, we just talked about, is not expressed in this sample. And so that value is 0. And so I see a lot of you guys added that up to give a value of 91 for this specific gene. So if you add up the expression values in counts for all of the isoforms for this specific gene, you get 91. And now I want you to compare that expected count value of the sum of those transcripts for this gene to the expected count value for that gene in our genes.results file. Going to be this line here. Is it the same? Is it different? Yeah, it's the same. Do you expect it to be the same? Yeah. Yeah, that's a good guess, Christina. Zinru, why do you expect it to be the same? You can unmute your mic.
Since this is by gene ID rather than transcript ID, it makes sense for you to sum all of them together based on the number of individual transcript IDs that we see in the transcript file.
Yeah, you're a little soft, but I was able to catch what you were saying. Yeah, that's exactly right. And so this gene effective count is going to be the sum of all the isoforms that are derived from that gene that are expressed in your sample. And so the value here, the expected count value of 91 for this particular gene 28, should be the sum of the expression of each one of the isoforms that are expressed from that gene. And that's what we were just looking at down here. So down here, we were looking at the expected count for each isoform. And so if you want to get the total count for that gene, you would add up the counts for each isoform that are expressed. So yeah, that's exactly right. And they should be the same. So you should be able to do that for any gene. And you should get the same value in both. Does that include non-canonical? So what do you mean by non-canonical, Felix?
So you said, I forget if it was star or RSM, finds splice transcripts that were not annotated before, novel.
Yeah.
Novel. So would those be included in these counts somehow, or how does that work?
Right. So no, these are only counting the annotated. So they're only counting the annotated transcripts and annotated genes. So the ones that aren't annotated that were discovered as novel and novel splice isoforms are not going to be counted here. Good question. Let's actually look at that because we saw some differences here, might have been. So count, I think it should be the same though, 869. Yeah, 869. Yeah, so that's not taken into consideration in here. So it's only considering the total being of the annotated. Good question. That brings us to the end of generating counts. Before we put all these together, also, can you pipe the output of Unix head into pandas here? That is a question for Lauren.
Good question. I think you may be able to save the output of Unix head to a variable that is accessible by Python, and then access it by Python in the next cell. So I don't think you can directly pipe it, like have Unix head and pandas in the same cell. but I think you may be able to have variables transfer between. Why don't you try it and see and let us know? Oh, you sounded so confident. Pandas should be imported. Well, at least it will be after you guys run the last cell.
Yeah.
Use it in the last cell. I will see if I can do it, Felix, and let you know in a few minutes.
So, Lauren, if you could do it when we transfer over to you, maybe that's the first thing that you can show before you get into your Jupyter notebook.
Sure.
Okay, good questions, guys. Okay, so our next step, now that we have counts for everything, that way the right column is aligned to the right number. The right column is aligned to the right number. It's just that, you know, it looks weird when you have it like this. So you're saying that the pandas function will let you align it better?
Amanda, you should do more coding in Python.
I know, guys. I don't know how to code well at all, really. Remember, I'm all self-taught. I learn what I have to do what I have to do.
Once you do pandas, you never go back to this.
I know. Lauren is like aching to transfer everything that I do into Python. And actually, a lot of the functions that we do, especially the R stuff, every day there's more and more packages in Python that allow you to do like everything we do in R with bioinformatics. So I envision that the future of bioinformatics is Python. So you guys are ahead of the curve learning Python, and I need to catch up. So Jonathan, you had a couple of commands there that you said work. So did you just add those in a separate code block, Jonathan?
So I haven't tried it yet, but in my intuition, this should work.
You're just throwing things out there without even testing them?
I will. Not quite everything ran, but if you were to copy your current code block and replace, trying to think if that would work, because I don't know how the RSEM output is getting interpreted.
Okay, yeah. So Lauren and Jonathan, both of you guys play around with that, and if you're able to get it working, then we'll have you share your results.
Oh, yeah. Jonathan, yeah, what you're saying will definitely work because this is a Python notebook, so you can just read in the RSEM files as tab separated files using Pandas read CSV function. But what I was curious about was whether you could actually convert between Unix output and Pandas output.
So you're saying, Lauren, if you just import Pandas and then do this and have this be our genes.results, but can we pull that variable though, the path variable here, or should we write it out?
You can, because it was defined in Python, but it
was defined in Python, but would you call it with a $?
Yeah. Why don't you I'll do this and I can show.
Okay, I'll get rid of that for now then. So we'll have Lauren, when she starts, she'll show how to look at this in Pandas, so that way everything's more prettily aligned in the output. I think I made up a word. Okay. Any other questions for generating genes.results? Kevin, you say that like I'm an expert R coder. That's not true either. That's why Lauren's teaching the R part. I'm really good at Unix. Let's see. Any other questions about generating counts data in RSEM, or any question about the RSEM outputs? Awesome. Okay. So now basically at this point, and again, I know we only did this ourselves for just the one sample, but I went ahead and ran the same commands that you guys ran just for all the samples, so that way we have all the data. And again, those are stored in those path variables that we created. Oh, actually, this ran because I kept mine running. Did everyone else's variables run okay, I assume? If you guys kept your Jupyter notebooks up, they should still be stored. So yeah, seems to be cool. So if for whatever reason you got an error here, a path error, you would just have to go up and rerun where we defined all the paths. So let's move on and let's put all of these count data together now. So remember, do you guys remember which file we use of the 2 counts files from RSEM for differential gene expression? You type it in the chat. So which of those 2 counts files from RSEM are we going to use for transcript? So it's actually the opposite, Diksha. So when we do differential gene expression, we're looking at changes in expression for genes. So we're kind of encompassing all isoforms and not specifically looking at differential expression in isoforms, but you could do that if you wanted to. So for our purposes, we're just going to look at all the genes. And so we're going to be importing the information from that genes.results file. And so there's a nice function in R called tax import that will allow you to pull in your genes.results files into a nice table in R directly. But since we're in a Python system right now, Lauren here had made this little Python script for us. So that way we can combine all of those raw counts data or the expected count data from the RSEM calculation for the genes.results files into a table. And so that's what this code is here. If you have any questions about this code, ask Lauren. But you type your questions in the chat and I'll just have Lauren answer if you guys have any questions about the code that she has in here. But from that initial survey, it seems like a lot of you guys have Python experience. And Dr. Heller said that a lot of you guys should have had at least 1 semester of Python. Okay, cool. So yeah, so this little code here is basically, it's written to pull the expected count value. So again, this 5th column here from the genes.results file for each gene for every sample. And so let's go ahead and run this code block. So it should finish pretty quickly. Hopefully you're able to do it in pandas. Is there anyone who hasn't finished running this code block? So once it's complete, you'll have a number here on the left in these brackets. Okay, so I assume all you guys ran it. Let's take a look at the table that we just created now. So go ahead and run your head command. And you should see the table that was created. So I have a few questions based on this table. So as you can see here, we now have in this first column is going to be our gene IDs, because again, we pulled this from the genes.results file. So we're looking at gene expression. And so we have our gene IDs here, and then across our column headers, each column is a different sample. And so remember, these were all, of course, aligned to the same reference genome, counted using the same reference being mouse, because these are all mouse-derived samples. And so it'll have the same list of genes, right? So all the mouse genes should be listed here on the far left column. And then each column represents the count data from each sample. So we're just compiling that that expected count for the from the genes.results file. And then just because you guys calculated it, remember we were looking at flight rep 1 when we were looking at those individual files. And if you guys remember, you calculated the expected count based on isoforms and then compared it to the genes.results file for this ensemble ID 28. And what you guys saw was an expected count of 91. So this is just a nice sanity check to make sure that you're getting the right counts associated with the right samples for the right genes. And so what we do when I do a V&V, so I verify and validate all of the output files before publishing them on our data repository. And how I verify that is I just pick a gene or two and get the values from all samples in this table, and I just pull that same information out of the genes.result file. So I pull this column 5 for each sample and just make sure those values match up. And so once you've confirmed that indeed you put all the right data together in the full table, let's go ahead and answer these questions. And so the first question is, how many genes are shown here using the now Python head function? You need, what was the command for checking the shape of counts again? What does that mean, the shape of counts?
In what language?
Number of rows and columns? Oh, that was in R. We were doing that, right? The number of the dimensions. That was the dim function. In Python, it'll be different.
Yeah, so dim is analogous to dot shape for pandas in Python.
Yeah, and so good call there. Yeah, Zinra pointed that out too. Counts.shape. I think you need open close parentheses, no, for that at the end. And then also noting here that Lauren had stored the table in a variable called counts. So that's why we called it the counts.head. Mice have that many genes. So
Yeah,
yeah, a lot of you guys said, oh, go ahead, Lauren.
What do they even need 55,000 genes for?
So yes, it is 5 genes that we are seeing in the head here. If you guys do that counts.shape, sounds like that worked for you guys. I'll try it on my end too. Oh, that didn't work for me.
Yeah, you just don't need the parentheses.
See, guys, look at me learning to code in Python already. Thank you. Yeah, 55,000 genes, 12 sample columns, right? That makes sense. Yeah, so keep in mind though, remember when we were diving into that GTF annotation file, some of those genes are repeated more than once. So I think for mice, there's around 30,000 annotated genes. And I think that's roughly the same for humans. And when you're talking about the fact that we have 3 trillion base pairs, only having 55,000 genes, I would say, why don't we have more? But yeah, some of those are duplicates, so there's not 55,000 annotated genes total. There's actually fewer than that. It's around 30,000 for both mice and human. We're remarkably similar to mice, just to humble everyone.
Is that because they're duplicated, or because some of the 55,000 just aren't annotated?
Felix was looking at the total number of genes here. So these genes are coming from our annotation file. And so those are only annotated genes. And remember when we were looking at the GTF file, we saw that there were duplicated lines for genes. So there was the same gene represented on more than one line.
Yes. I thought you were saying the reason, but you were saying there are actually 30,000.
Yeah, I'm pretty sure there are around 30,000 annotated genes for mice.
So some of these 55 aren't annotated in some way or another?
Some of them are duplicated, I'm pretty sure. I think we do a filtering step for that in the DC2 script, but I'll double check on that to make sure that I'm not lying to you guys. Yeah, so we see 55,000 rows, 12 columns. So when do you mean duplicated? Meaning that it's the same gene.
So here, gene 1 and 3 or 1 and 28, for instance, might be the same gene.
Right.
Is that not a problem then? How?
Yeah, I'll double check that and get back to you guys, but that's just from what I remember. But I see how that's confusing. So let me double check that and get back to you guys before you believe me.
Because if that's true, that means, yeah, if one had 800 for one sample and 0 for 28, even though they're the same gene, and then it was flipped for sample 2, then you would say they're differentially expressed, even though they're not. Wait, does that make sense? So imagine 1 and 28 are the same gene, and then you have 800 counts in one for sample 1 and 0 counts of 28 for sample 1 as well. But then for sample 2, you have 0 counts of 1 and 800 counts of 28. So there is no difference. But
right. So if it were to be the same gene, you would anticipate seeing roughly the same number of counts. And I mean that so because when people, right, so genes are discovered. And so basically people do a ton of molecular biology and say, oh, let's see what's expressed here. Ooh, I think I found a novel gene. Here's the sequence. Let me see what this function is. And then they make up a name for that gene and then they publish that information, right? And then they submit that data to a database and they say, hey, I discovered this gene. This is the location of it on the relative to the reference genome. This is the function that it does, but that could happen multiple times, right? So researchers are doing research all the time. Researchers are working on similar things all the time. And so someone like in a lab here in America could discover a gene and a function and submit it to a database. And then that's ingested into a new version release of Ensemble. And around the same time, someone in Korea in a lab in Korea could have been working on something similar, discovered a gene, named it something differently, and then submitted it to a database that's incorporated into the next version. And then it turns out that those genes that those two different labs discovered are actually the same gene. And so then it has to get reconciled with a new version of Ensemble. So that's kind of what I mean by that there could be duplicates of the same gene.
So you mean that we might not necessarily know right now that there are duplicates, but
right. So
where did the 30,000 number come from?
I've looked it up a few times how many annotated genes there are in humans and mice.
I see.
Yeah. So I'll double check that, and so that's why we're gonna transfer these gene IDs into common names or gene names. So once it's reconciled, if like a new gene is imported into a database, it's given like that database's number. And so it would be like an Ensemble number here. And then once it's figured out that might be, there's two Ensemble numbers that represent actually the same gene, then there will be a decision made for what gene name is going to be used moving forward. And so when we do our annotations and map to certain gene names, then we can filter out based on the gene names and get rid of duplicates. So I will confirm all this tonight and either confirm what I'm saying or correct what I'm saying tomorrow. Good questions. I'm glad you brought all this stuff up. And so let's continue through with these questions for this table. So which of the genes shown are not expressed in any sample? Yeah. So that ensemble ID gene ending in the number 3, you see that it's 0 in all samples. So this gene just is not expressed in any of these mice. Again, this is in the soleus muscle tissue. So just to remind everyone, bring everyone back to the start of the experiment. So this is from the calf muscle, one of the calf muscles, so one of the quadriceps, known as the soleus muscle, and that muscle happens to be one of the muscles that's most affected by weightlessness or microgravity. So that's why this is, I picked this data set, is because it's pretty interesting. So yeah, this gene here is not expressed in any soleus muscle in any of these animals. And then this last question I have here is, which sample has the most reads aligning to the gene ending in 28, so aligning to this gene, and which sample has the least? Yeah, flight rep 5, Diksha has the most. What about the least? Which sample has the least? Flight rep 3. Yeah, so big in the same group, right? So again, these, want to keep in mind, these are raw counts, so we still do have to go through and normalize, but we can already start to see that some genes we see differences in the levels of expression between the different groups and even between the different samples, but of course we have to go back and normalize that. And so we now have a table containing the raw counts for all genes for all samples in our study. We're going to use this table to perform our downstream analyses, including differential gene expression and principal component analysis, also gene set enrichment, and generate respective visualizations. You guys are going to be doing all of that with Lauren. I want to check our break times. We have a break coming up at 2:10, so I should be able to finish my lecture before the break, and then after the break, I'll hand you guys off to Lauren. So any other questions about anything in this FastQ to Counts Jupyter notebook? This brings us to the end of it. Yeah, pretty big accomplishment. Okay, let's hop back into the lecture then, and I just have about 6 slides for you. Question?
You're trying to figure out how to convert the isoform results table into pandas, right? I think I have the code for that.
Oh, to combine this for the isoform table?
Oh no, from was it from section 6? We're trying to figure out how to convert it into
yeah.
Yeah, I got the code for that.
Awesome, put it in the chat. Okay, so you did import it to a variable. Cool.
And it is a lot cleaner. It's easier to read too.
All right. I know. It's on my to-do list. If I ever have free time in my life, it's the first thing I'm doing. I've also talked about taking a sabbatical from working just so that I could learn how to code and stop doing this fake it till you make it game. Yeah, so that's what you were looking for, guys. Nice and pretty, aligned nicely. Thank you, Kevin, for putting that code in the chat. It definitely does make it a lot easier to see things. Cool. Okay, let's move on to these last 6 slides or so that I have before the break. So RNA-seq data analysis. So keeping track of where we are in our pipeline. So we've gone through, took the quality of our raw reads, we trimmed and filtered those raw reads and then checked the quality of that trim data. We then aligned our reads by creating a star index and then used the star program to align our reads. And then we looked at how well we aligned our reads. And then we just got done generating an RSEM index and then using RSEM to quantitate the number of reads aligned to each gene for each sample, and then compile that information from each sample into a raw counts table. So this, guys, is pretty much the end of data processing. And so this part of the pipeline up until here, once you get that counts table, that's pretty much what people consider is the data processing portion. So now we have a counts table that we could start playing around with and analyzing the data however we see fit. So this portion from here on out is more so considered analysis. So it's analyzing the data that we just processed. And so the first step in our analysis is going to be to normalize our counts data. So some of you guys had pointed out, like, what are we going to do because we have transcripts that are longer and we noticed that there were differences in read depth. So now we're going to talk about all that stuff and how we account for it. So I'm going to go over it briefly, and then Lauren's really going to dive into the statistics and stuff that are used in the normalization methods that we have. So I'll just do a quick overview of what we're going to be doing, and then we'll go to the break, and then we'll move on to Lauren's Jupyter Notebook and lecture. So we now have quantitative gene expression data. Are we ready to run differential expression analysis? Kind of already gave you this answer, so I'll just move it along. So not quite. So although we've counted the number of reads aligned to each gene, we have not accounted for differences in read depth. We haven't accounted for differences in gene length or differences in RNA composition. So why do you guys think this is necessary? And you guys just give me a general answer if you want, as opposed to something specific. So why do we got to account for these differences before we start comparing? Oh, I'll get to that. Yeah, easier comparison. So more, yeah, removing bias. That's what I'm looking for. Yeah, those factors affect the gene count and affect how we can compare our differential expression. So again, we don't want to be introducing any bias due to technical differences in our samples. So yeah, all good answers. So first, read depth. So I know we touched on this a little bit earlier, but just to put it in a pictorial representation here. And so let's say we have sample A here, which was sequenced at 100 million reads, and we have sample B here on the right that was sequenced at 50 million read depths. That means we have 100 million reads for sample A and only 50 million reads for sample B. Now when we go to align those reads to gene X here, gene Y, and gene Z, if we don't normalize for read depth, it's going to look like we have double the amount of reads for each one of these genes in sample A than sample B. But in reality, that is likely due to the fact that we just sequenced at double the sequencing depth in sample A. Right, so this then we would say, oh, these genes are double the amount in sample A than they are in sample B, and we would be likely wrong in saying that if we didn't account for those technical differences. So that's normalizing for read depth. Next is gene length. This is another one that we touched on a little bit earlier. And so looking at this example here, so let's say in sample A here, we have gene X and gene Y. You see gene X is quite a bit longer than gene Y. And so looking at the number of reads here, here we have gene X and gene Y have similar levels of expression, right? But if you go by the read counts, gene X has double the number of reads that are aligning to it than gene Y does. But that's just because gene X is longer. So one copy of this is when you go to chop up your DNA, you're likely going to have more little pieces for just one transcript than you will pieces for just one transcript of gene Y. And so if we want to compare expression of gene X and gene Y to each other within a sample, then we need to account for those differences in gene length. Next is accounting for RNA composition. And so this is dependent on how you're normalizing your data. So that's why you want to be really cognizant of it. So in this example here, what we can see in sample A is that we have gene X, gene Y, and gene Z. And then we also have this gene DE here that is a contamination. And in sample B here, again, we have gene X, gene Y, and gene Z, and it doesn't look like gene B or sample B has that contamination. So now if we're normalizing our number of counts based on the total number of counts in the sample. So now when we take the total number of counts here, total number of reads in sample A, and we take the expression of gene X, the number of reads aligning to gene X, and divide it by the total, and the number of reads aligning to gene Y divided by a total, and do that for gene X, Y, and Z, the relative expression of gene X, Y, and Z is going to be smaller now compared to sample B, but it's just because the total number of reads was more due to this contamination, right? So remember, we have that bias in the flow cell that smaller, shorter inserts are going to latch on more quickly. So you might have, if you have a contamination that happens to be a shorter read, then it's going to have more reads in sample A. So when you do this normalization, you could be skewing your data if you don't count for the RNA composition here. And so that's what we mean by RNA composition, is so when you're normalizing relative to the total, you need to take into account if we have things like large expression of a contaminant. So that's what I mean by composition. Questions first about those three types of normalizations and why we have to do them. No, makes sense? Awesome. Yeah, pictures really help. Okay. So now we're going to look, so which of these three factors, right? So we just discussed normalizing based on read depth. We discussed normalizing based on gene length. And we discussed normalizing based on RNA composition. So of those three things that we just discussed, which one of them do we need to absolutely account for when performing differential expression analysis? So when comparing expression between two different groups of samples. Write your answer in the chat. Guesses? What do you guys think? Which of those three things, read depth, gene length, and RNA composition, which of those three do we have to account for if we want to perform differential gene expression without introducing bias? All RNA composition. Any other guesses? Read depth, length. Okay, getting a mix of responses here. So let's think about this. Let's start first with read depth here. So read depth, the picture kind of tells you this answer for you. So in read depth, what we see here, right, is now if we want to compare expression of these genes in sample A to expression of these same genes in sample B, is it going to be a problem? Are we going to introduce bias if we don't account for read depth? Yeah, I'm seeing yeses. Yeah, for sure. So we're comparing, right, the expression of gene X in sample A to the expression of gene X in sample B. So if we have more reads in sample A than we do in sample B, by nature of having more of a sampling of the transcripts, we're going to capture more reads that are aligning to gene X. And so we will see that gene X is more expressed in sample A than sample B, and it's likely just because we have more reads. So we absolutely want to make sure that we account for read depth when comparing two groups. So yes. Now what about gene length here? So gene length, we have gene X, right, which is a certain length, and we have gene Y, which is a certain length. And now keep in mind that gene X is going to be that same length in sample A. Gene X is going to be the same length in sample B and sample C, and any sample we look at, as long as we're using the same reference genome. right? And so is it important to correct for gene length when we're doing comparisons for between samples? No, maybe not as important to read that. Yeah, you guys are getting it. No, yeah, for sure. So yeah, we don't actually need to, right? Because it's kind of like having that same error or same issue in everything equally. It kind of falls off when you're doing comparisons, right? So if we were to normalize this for gene length for gene X, right? So let's take the number of reads and divide it by the gene length. Right? So let's say the number of reads are 100, the length of gene X is 50, and so we divide number of reads aligning to gene X by 50 in sample A. Well, gene X is the same length in sample B, right? So then we take that number of reads and divide it by 50 in sample B, too, and then also in sample C, right? So you're dividing it by the same number in all samples. So when you're doing a comparison, right, that ratio of the total number of reads in sample A versus the total number of reads in sample B is going to be the same, even if you don't account for gene X, because you would take that number and divide it by the same number for all samples. Does that make sense? So when would be important to normalize for gene length? What kind of comparison, if you want to compare something, what comparison would require you to normalize for gene length? Any guesses? Yeah, Zinru, you got it. Yes, Hannah Lee, yes. So if you want to look at just sample A, let's say, and you want to know if gene X is more highly expressed or gene Y is more highly expressed, now you absolutely have to account for gene length, because now you're comparing within the same sample here. And so if we have 100 reads here mapping to gene X and 100 reads here mapping to gene Y, that does not mean that gene X and gene Y are equally expressed, because gene X is a lot longer, so the likelihood of you getting a read from even just one copy of this transcript is going to be a lot higher. So yeah, when you're doing within sample comparison of gene expression, you do need to account for gene length. Yeah. So what about RNA composition? What about this one? Is this something that is going to affect comparison of sample A and sample B in this example if we don't account for that? Yeah, Zinru's saying yes. Yes, so this is another one that, again, if we take a look at, so if we do our normalization here and we normalize based on total number of reads, and so we're normalizing all of these based on the total number of reads. If we are blind and don't consider the composition of the RNA that's there, so don't consider or check to look for if there's a contaminant or something, that we have a ton of reads. And we saw, remember, when we were looking at our duplication levels, so those really high-end duplication levels, which likely indicate contamination. If we're not accounting for that, then we're going to be normalizing based on the total number of reads here, and based on the total number of reads in sample B, but sample B's total number of reads is less than sample A, but it's just because sample A has this contaminant that sample B doesn't have. And so when we normalize based on the total number of reads here, what would happen is that we would then reduce the relative expression of gene X and gene Y more so in sample A than we would in sample B. And so that would be artificial, because that's not showing the true expression of these genes relative to one another when you normalize for all reads without considering RNA composition. So getting back to this question here, which of the three factors that we discussed need to be accounted for when performing differential gene expression? It's going to be both read depth and RNA composition. So whatever normalization tool we use, we want to make sure that we're correcting for both of those things. Okay. Are you guys okay if I go 5 minutes over into the break? I'll still give you a 20-minute break. We'll just start at 5 minutes later. Is there anyone who has to stop at 10 after? Okay, how do we know the composition for the RNA? Good question, Rashi, good question. Unless we know like which sequences are contaminants, that would probably be difficult to determine. And so different tools, independent of knowing what the contaminant is, it will use different ways to normalize based on read depth rather than just taking the total number of reads. So rather than looking at the total number of reads, it could look to say what is the average number of reads aligning to each gene. And so when it looks at the average number of reads aligning to each gene, if there are some genes that are have a large number of reads aligning to it, you might not want to include that in your normalization. And so you might instead want to look for what is the median number of reads that are being expressed in the genes to do your normalization. So that's kind of how you account for your composition of RNA without actually knowing exactly what the contaminants could be. And Lauren's going to talk more about how that's done in the context of different statistics used for the tool that we use to do this normalization. So she'll get into that a little bit more, and hopefully that'll clear things up a little bit more. Okay, so there are, just like anything else, a bunch of different tools in a bunch of different ways you could use to normalize your count data. In this, guys, what you choose to normalize your data and how you choose to normalize your data has the greatest effect on what your differential expression analysis is. So remember how I told you that there's a bunch of references, and I'm sorry I haven't put those in the Google Drive yet, but I will, of people comparing using different tools and setting up different pipelines to do their RNA-seq data processing and analysis. When they do the tool comparison, the thing that causes the most differences in their output is the type of normalization that they do. So this is a big one. This was a big area of contingency for us when deciding on the pipeline too. And so just a couple of normalization methods here. I'm going to go over these fairly quickly, and you guys can read more about them. And if you have any questions tomorrow, you can bring them to me, because we're really just going to focus on the normalization method that we use in GeneLab. But so just to let you guys know, so counts per million is a type of normalization method used. Limavoom is one of the tools that uses counts per million. And so the description for how that is the total reads in each sample is divided by 1,000,000, and that creates a scaling factor. And then the gene counts are divided by that per million scaling factor. And so, as we just talked about, that'll account for differences in sequencing depth, right? Because we're taking the total, taking into account the read depth here by dividing by that per million scaling factor. But we don't take into account RNA composition doing that, right? So that was just the example that I showed you with what not to do with normalization for when you're normalizing for read depth, to making sure that you can do differential expression comparison. And so to compare counts, so the uses of this is to compare counts between replicates of the same group. So you wouldn't want to use this for within sample comparisons or for differential expression analysis for the reasons that we talked about. And then so, yeah, Lauren, so I just asked everyone if it was okay to hold on for 5 more minutes. So I'll make sure I stop it at 2:15. I just have one more slide. Well, maybe 3 more slides after this, I think, but they'll be quick. And so the next one here, the next type of normalization method is reads per fragments per kilobase million. or RPKM or FBKM. So this is one of the normalized counts that RSEM generates. You guys remember that column name? And so counts per million values in this case. So first counts per million is calculated using the same method as described here. And then that counts per million value is divided by the length of the gene in kilobases. And so in this method, we're going to account for both sequencing depth and gene length. And you can, when you do this, you can then compare gene counts between genes within a sample. But again, because we're not taking into account that RNA composition, it's not advisable to use this for between sample comparisons or for differential expression analysis. Could you use a RNA spike-in for normalization? Oof, that is a big discussion that we constantly have on GeneLab. Can you hold that and we'll talk about it after the break? So yes and no is the answer to your question. So the next type of normalization that that people do is transcripts per kilobase million, or TPM. And so this is the other type of normalization counts that RSEM spits out. This is also the type of normalization that Salmon and Callisto use. And so this is very similar to the reads per kilobase million or the fragments per kilobase million that I just talked about, except the order of operation that it does is kind of reversed. So first, the read counts are divided by the length of the gene in kilobases. And then the sum of all of those read per kilobase values for each sample is divided by 1,000,000 to give you that per million scaling factor. And then those read per kilobase are then divided by that per million scaling factor. So just the order of operations for this is opposite of what you do to generate this. And again, so it accounts for the same thing, sequencing depth and gene length. And uses here is to compare gene counts within a sample or between samples of the same group. And again, not so much for differential expression analysis. So we are at 2:15. I did not finish getting through my talk, but I will finish right after we break because I want to make sure you guys get your break in. So meet back here at 2:35. I will wrap this up within 10 minutes and then we will hand you over to Lauren.